Hey guys, welcome back to DNA. Now, in a future chapter, we are going to be talking about how to make brand new cells. Yes, this is important right now, so quick rundown. One cell goes through a big long process we're going to talk about, but eventually splits into two cells. Ignore the ink on my hand, I don't know where that came from. Two cells. In order to have two cells, we need two strands of DNA. So that means rewind some point over here in the beginning, we need to make new DNA. That's right, we need to start off already with a second set of DNA so this whole thing can happen and make a new cell. So how do we make DNA? Now that is the question. And you remember my board from last time, it's altered a bit, all right? Because it's already in the process of making new DNA. Now there are two things you're gonna have to remember for this. One, the nucleotide. That's right, the nucleotide, that's what the DNA is made out of. Nucleotides are just floating around, new ones here in green, right? I drew them green this time. And they're just floating around inside your nucleus. The nucleus of the cell, which is where you keep your DNA, remember that? That is also where you're going to make your DNA. So all of these nucleotides are just floating around. You made them from all the food you eat, okay? Chapter 2 stuff. So they're there ready to be used. In that nucleotide, bases are going to pair up with other bases. And the phosphates attach to sugars, which will attach to another phosphate sugar, to another phosphate sugar, and that's how you get the chain. Phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, all the way down. And the nucleotides are made out of carbon, which we've numbered. There's a carbon here, we call it 1'. And 2', 3', 4', 5'. But what we care about is 5' and 3', right? Because that is the direction of DNA, and that is the direction that you build your DNA in the 5' to 3' section. So this side, the DNA is going to be built in this direction, assembled on this side. Oh, camera. The DNA is going to be built in this direction, and then on this side, 5' to 3', the DNA is going to be built in this direction. Second thing you have to remember is bonds. That's right, we talk about two different bonds, covalent bonds and hydrogen bonds. Covalent bonds are what's holding the chain together, and this is really tough to break, all right? You pull on this and nothing is going to happen. However, hydrogen bonds, super easy to break, and that's what holds the bases together. Remember that? So these bases break easy, and that's what we want, to open up the DNA. We're going to unzip it like a zipper. So there is a protein that's going to do that. And remember, proteins do everything. So there's a protein that does that, and we call it helicase. Just riding up here like a zipper, and it is breaking the bases apart as you go. And while that is happening, the DNA is being assembled. These extra pieces floating around are now going to start base pairing. Oh, third thing you have to remember, the base pair rules. I have a G at the bottom. What pairs with G? That's right, a C. And then a C pairs with, obviously, a G. What's A pair with? A T. You remember those rules? A's with T's, C's with G's? Good, 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 good. Got it. All right, so let's start assembling. Now that this is opened up, it's musical chairs. Everyone for themselves. Quickly, find a base pair. And so as this protein starts riding up and unzipping, these extra pieces start filling in the gap. And now we got new base pairs. So we used to have this G with this C, and now the G's with this C. So it used to be GC, and it's still GC. It used to be C and a G, and it's still C and a G. It used to be A and a T, and it's still A and a T. So the code did not change because of the base pair rules. But we're not done yet. We still got this side to work on. Remember what I said? It builds in the 5' to 3' direction. From the 5' to 3', we started at the 5', we built toward the 3'. So that means we had to start with the 5', and build toward the 3', right? So as this guy started, he started down here, right? So as this guy started down here, he started working up, and then we started adding bases right here. And we started building here, and we worked from 5' to 3'. And 5' up here, 3' down here, going in this direction, all while this guy was still going up. So now we had to back up and then start here again and go back down. So this side was one smooth build. This side's being built in chunks. This way, and then come back up in this way. So again, we started here, it went this way, and then we came up and built this way, and then we come back up and build this way again. And now look at the code. This side is AT, AT, TA, CG, GC. Over here, AT, AT, TA, CG, CG, or GC. It's the same code. We are building two completely identical strands of DNA. And when this whole thing is done, that's what we'll have, 2 strands of DNA. So back here, that happens here, and then a whole nother chapter of information, and we end up with 2 cells, and each one gets a strand of DNA, and they will be identical. So let's jump into the actual notes so you can write all this stuff down. So DNA replication, taking 1 strand of DNA and turning it into 2. As I said, the nucleotide, the sugar has 5 carbons, 1, 2, 3, 4. Okay, the 5th one's like in between the sugar and the phosphate, but whatever. We care about the 5' and the 3' carbons. And if I'm going too fast, by the way, feel free to pause and write things down. We have different kinds of bonds holding them together. The phosphates to the sugars, and then sugar to the phosphate. Look at, they're not connected. Yes, they are. They have what we call a covalent bond, where atoms share electrons. So an atom in this phosphate and an atom in this sugar are holding on to the same electron right here, which is an extremely strong bond. These two will not break apart. And that happens all the way down here and all the way up here. From base to base, on the other hand, they are not sharing electrons. They're more like magnetically drawn to each other. Or just like how the water molecules have a polar nature, and that creates a cohesive attraction to each other, kind of like magnets, and that's what's happening here. They are drawn to each other like magnets, so it's not a physical connection like it is here. They're just drawn to each other. And this is a very easy bond to break. Now here's a new word for you. I've already described it though. Anti-parallel. We know what parallel means, where 2 things are right next to each other. Like these pencils. They are parallel to each other. They are going in the exact same direction. Now, what does anti-parallel mean? Does it mean going like this? No. That's perpendicular. Remember that one? No. Anti-parallel is still parallel, just going in the opposite direction. This one's pointing upwards, this one's pointing downwards. They're still parallel to each other, but in opposite directions. So this side is in the 5', that's actually a whole lot of numbers. Let me get rid of some of them. This side is in the 5' to 3' direction, and this side is 3' to 5'. We call 5' to 3' the leading strand, and the 3 to 5 the lagging strand. Why do we call that lagging? Because if you remember, this side had to keep doing it in chunks. This one was one steady strand. That was the leading strand. They went 5' to 3'. The 3' to 5' was in chunks. It went this way and then went this way and then went this way. That was slower. So it was, if this was a race, this side was lagging behind. See? Lagging, the lagging strand. Let me tell you a little history lesson. Once upon a time, people didn't know what I showed you on the board. Yes, that was a complete mystery because we didn't have microscopes, we didn't understand the chemistry or blah, blah, blah reasons. People didn't know how DNA replicated. So they had models, they had hypotheses, they took guesses, all right? And there were three leading guesses that they cared about. One was called the conservative model of replication. You know what conserve means? To conserve means to save. So we got the original DNA, and by some magic means of doing things, your body, they thought your body just made another thing of DNA. This was wrong. Why am I showing you a wrong thing? Because I want to show you that science is a process. You're going to get a lot of things wrong before you get it right. Then they had another idea. They went with Chargaff's base pairing idea. The A's and T's are pairs, so there should be equal amounts, blah, blah. So they thought the dispersive model, the DNA rips into pieces, and then they fill in the gaps with the base pairs. Okay, that makes sense, but look at the chunks of the original here. They're scattered. They're not in any particular order, so it doesn't explain how to keep your DNA in the correct order. Remember, it's a message. This is a language. These bases are letters in an alphabet. If you rearrange them, just like letters in English or Spanish, if you rearrange them, you make different words. You do not want to rearrange the message. So the final idea was what I showed you on the board. We call it semi-conservative. Kind of conserving. We take the two sides and separate them. So one strand is over here, and the other strand is over here. So we conserved or saved the original individual strands without breaking the nucleotides apart. And that's what I showed you right here. Yeah. The original strand is still intact because the phosphates and the sugars would not let go of each other. That's the semi-conservative. This side was conserved, this side was conserved, but it's only semi-conserved because we broke it here. Remember that your body has tools, and we call these tools enzymes. That's right, enzymes, again, from chapter 2. Man, I keep dipping into chapter 2, don't I? Enzymes are proteins that have a job to do, and they are going to help chemical reactions take place. This is a chemical reaction. Molecules are just another fancy word for chemicals, and they are reacting with each other, chemical reaction. And this is an enzyme. This enzyme is called helicase. Putting a big old H there for helicase. Its job is to unzip the DNA. So I drew it like a zipper. And then we have another enzyme I did not draw here because I was lazy. It is called polymerase. And polymerase's job, once these bases get together, polymerase's job is to make sure that they actually stick together. And to take this individual nucleotide and this individual nucleotide and this one and lock them together with the covalent bonds, making them one big molecule. So polymerase is going to be on the leading strand, going to be following helicase. As helicase unzips and they assemble in the 5' to 3' following the helicase, and polymerase is going to be following all of that, attaching them, going up. And then on this side to be a little different because they built this way. He goes down, lifts up, and then goes down, lifts up, and goes down. You get the idea. Polymerase. By the way, you put ace at the end of a word, we're talking about an enzyme. Polymer, ace, polymerase. It is an enzyme that builds a polymer. Here are the words, you can write them down. Helicase is an enzyme that unzips the bases. Polymerase is an enzyme that attaches the nucleotides to each other. You can pause this and finish writing it. I'm moving on. So here's the whole process. This time it's going to be in motion though, all right, instead of just on my whiteboard. So follow along. And again, if you need to write this down, feel free to pause. I'm not going to be waiting for you. Step 1, we take our DNA right here. Just Actually, much bigger, whatever. And then helicase is going to come in. Today, helicase is played by Pac-Man. On the board, he ate upward. Here, he ate downward. Doesn't matter. He went in the 5' to 3' direction. That's what matters. And now, step 2, these old strands are going to act as a template for the new DNA molecules that are just floating around. What's a template mean? It's what you use to trace if you're not a good drawer. All right, that's a template. This is the template to make new DNA. Brand new nucleotides, with the help of polymerase, are going to attach following base pair rules. This is a T. It used to be paired with an A because T's and A's go together. So an A is going to pair up. And once they're together, that polymerase is going to attach this nucleotide to the next one that comes in. And now this phosphate and this sugar are together. And by the way, G paired with the C. And then going down the line, because helicase went in this direction, polymerase on the leading strand will follow behind. All in one go. So let's rewind that. All in one quick motion. The other side, the 3' to 5' direction, is going to be opposite. Remember, it can only go 5 to 3, which is the wrong direction. So it is going to attach these guys and then jump off and then add more and then jump off and then add more and then jump off and add more. So this guy takes longer, which is why it's called the lagging strand. It's lagging behind. And they're all still connected with covalent bonds. And step 4 isn't even a step. Step 4 is you're done now. Replication is over, and we now have 2 completely identical models of DNA. The original said A and a T. Now the 2 new ones say A and T. And then the original said C and G, so the 2 new ones say C and G. All the way down, they're completely identical. And like we talked about before, they're both in this cell. So in our future chapter that we're not there yet, called mitosis, this cell will go through a big long process of separating those 2 strands of DNA. And then once they're separate, one goes in this cell, one goes in that cell. Wait, that doesn't look like DNA. That's weird. And what are they separating? Yes, guys, that is DNA, but I didn't tell you something because that was the next thing. And you see in the picture, all of that DNA is a hot mess. So when they have to separate, it's much easier to organize them. So your body's going to braid that DNA up. So you're going to cut your DNA into chunks and then braid those chunks up. We call those chunks chromosomes. Chromosomes, a coiled strand of DNA and proteins. Right, so just like when you're trying to braid your hair, you need some like twist ties or something, rubber bands, something to keep it twisted up, keep it braided. Well, we're going to need proteins. That's what the yellow balls are, by the way. These are proteins called histones. They are going to be used to keep the DNA all braided up. Then we're going to take that braided up piece of DNA and braid all of them up together. So it's braided around the histones, and then we braid all of those braids together. And then we take this braided braid and you guessed it, we're going to braid that up. And that's what a chromosome is. When you are made, yes, made, as in your mom and your dad got together and made you, your mom gave you 23 chromosomes and your dad gave you 23 chromosomes. So you have 23 pairs of chromosomes. That's 23 and 23, 46. Human beings have 46 chromosomes. That's one strand of DNA. All right? Pieces. You had 23 pieces from mom, 23 pieces from dad. You put those pieces together and you make your complete strand of DNA. When it's time to make new cells, your complete strand of DNA is going to cut itself back into those chromosomes again. And because you went through all of this, you now have twice as many chromosomes. That's why this blue one is paired up. And then there's 2 orange ones, 2 green ones, 2 purple ones, 2 red ones. So you get the idea. There's 2 of every chromosome. And a whole chapter about splitting them apart. So your 2 strands of DNA, one on this side, one on this side, and they end up going into their own cell. And that's how you make your DNA to make new cells. So just recap. The chromosome is before replication, we can say it's a single coiled bundle. All right, this is actually a lie because it won't coil up before replication. Just go with me on this one. After replication, you have 2 sides, and that's why chromosomes look like an X. And like I showed you in my poster, this side will go to one cell, and this side will go to the other cell. And then they will untwist inside those cells and go back to normal. And that is how you make brand new DNA, so that you can make brand new cells, so that you can replace the cells I just killed. Yeah, I just killed some skin cells. We'll revisit that in a future chapter on mitosis, but for now, I just want you to remember the how to make DNA parts of it. That's it for the DNA portion of the notes, but no, we're not done yet. We still have to talk about RNA in a future set of notes, so this chapter is still going on, right? But for now, study up on DNA, see if I left any work for you, and go do that. Good luck, guys. I'll see you in class.